So last time we finished up talking about the viruses, and then we started talking about genes and operons and DNA replication. So today we're going to finish up the chapter 7, but so these questions are kind of a good primer for finishing up our discussion about replication. So can anybody tell me what DNA does? Yep. Does anybody remember what the other name for the DNA primase is? Yep, DNAG. All right, so like I said, today we're going to finish up with replication, and then we'll move on. So we'll talk about plasmids because that's the other kind of DNA in your genomic content. And we'll talk about some of the tools that have come out from some of these different mechanisms that we've been talking about. So how restriction enzymes work, and then how the sequencing technologies work. And then after we get through that, I got a few requests for topics to go over, so I'll go over some of that stuff for the exam. Okay, so just back up a little bit and just kind of remind you broadly what happens with initiation. So again, so you have your origin sequence, and a few things need to happen, right? So the origin has to be methylated correctly. There has to be enough of this DNA A ATP complex around. So once those things happen and you don't have any of the CK kind of still on there, once you have enough concentration of the DNA ATP, they will bind to these regions here, and depending on the species will depend on how many of those actually required to bind. So that DNA binding will initiate some of the strand melting, and then we'll start loading all the different things that we need on there, right? So we need DNA B, but to actually get DNA B to get on there, you actually need DNA C. So B and C come together, and then C is actually able to load that onto where the A has melted the strands. Once C does its job, it leaves. And then, so now what we can have is the DNAG, the primase, come and bind on there. And what that will do is it will create a RNA primer, right? So we remember that DNA polymerase can't actually just start synthesizing new DNA from nothing. It needs that 3' hydroxyl group for it to start adding bases to. So you get the DNAG binding and the primer starting, but now you want to bring in the actual polymerase molecules, and so you have this sliding clamp and the clamp loader. So it actually loads 2 separate DNA polymerase molecules, and you'll have one of those at each end of the fork, and so those will load on, and then you can finally start doing some replication. We left off kind of in the middle of elongation, so I'll go over elongation again. Like we mentioned last time, so DNA actually contains 5 different polymerases, but only pol 1 and pol 5 are directly involved in their replication. So DNA pol 3 is the main one that you can think of, and it has several different subunits. So not only do we have several different polymerases, within the polymerase there's multiple subunits. The alpha subunit is what's involved in the DNA synthesis. The epsilon subunit, DNAQ, is used for proofreading. So, it can correct mistakes. It'll kind of go and double check and make sure that it's putting the correct base there. And then they have other subunits that are, again, involved in fidelity, so making sure that the accuracy is right, and then processivity, so making sure that that whole thing doesn't fall off too soon. All right, so we also talked about how there is a leading strand and a lagging strand, and this comes about because of the fact that you can only go 5' to 3'. And so when you're, and it's happening bidirectionally. So in one direction, it's going to be fine. It's just going to go 5' to 3'. In the other direction, if you were to kind of just go along the circle, it's actually going in the wrong way. And so what it has to do is kind of jump ahead, put some primers, and have it go back the other way, and keep doing that again. And so those are what are called the Okazaki fragments. And so you will create these larger single-stranded regions, and what actually needs to happen to protect that from, so when you see single-stranded DNA around that, it's actually in trouble for being cleaved, and so you can actually destroy that. And so the cells have these single-stranded DNA binding proteins that kind of just sit on the single-stranded DNA and protect it while it's single-stranded. And so this whole process happens about every 1,000 bases. And so at each time, you need to have a new RNA primer made by DNAG. Okay, so what that leads now is when you have the lagging strand, you have all these patches where now that there are a bunch of these RNA primers in there. So you actually want to replace those with DNA. You don't want it to be an RNA-DNA hybrid. And so there are a couple of ways to do that. So in DNA, POL1 has some 5' to 3' exonuclease activity, so it can actually come through and take out the RNA. Or there's an actual specific RNase called RNase H that only cleaves RNA when it's in this RNA-DNA hybrid. So if it sees RNA bound to DNA, it'll go bind that and it'll chew up just the RNA strand. It won't touch the DNA strand. And so it won't touch single-stranded RNA, and it won't touch double-stranded RNA, only the hybrid. So we have the RNase H comes here, and it clears off the RNA primer. And then POL1 will come in and fill in the gap. So POL1 doesn't have the ability to join the 3' hydroxyl with the 5' phosphate at the very end. And so another enzyme is actually needed to be able to link that, make that final linkage. And so that's the DNA ligase. Okay, so that was elongation. So now coming up to termination. So termination comes about based on these TER sites. And so in E. coli, there are actually 10 sites. So, they're kind of repetitive just to make sure that it actually stops. You don't want it to keep going. And so it builds in kind of these extra safety sequences just to make sure that it actually stops. And then, so because one is going this way and one is going this way, it'll have 2 sets of sequences so that one will read in the clockwise direction, one will read in the counterclockwise direction. There's also these TUS sequences. So these are the terminus utilization substance. So these bind to the TER sequences, and then it stops the DNA B helicase activity. So if you remember way back from the initiation, DNA B is the one that has the helicase, so it's going along ahead of the thing and opening up all the strands. And so when it sees these, when it hits these TUS molecules, it'll stop the helicase activity. But now, in the end, once you've replicated and you terminated, you end up having 2 strands that are actually linked together just because of the way that they were started, that they're interconnected. So you need to be able to separate the 2 different chromosomes. And so this XERC and XERD actually go ahead and cut those, make a cut and pass them through, and then rejoin them together. So now that you'll have the 2 separate genomes. I think I posted this. If not, I will double check this after class. There is a video that goes through the whole process. I highly encourage you to watch the video for this replication. And then again, this is just an overview, like a high-level view of what the whole process looks like. So starting with the chromosome, you'll start your initiation, and you'll start having the bidirectional synthesis. And this kind of shows as the fork moves along how the process goes. And you see in the lagging strand, you see all the different fragments here. And then again at the end, it terminates at the end. So like I said, watch the video. I will double check and make sure that I actually posted that. But any questions about replication? Okay, so the other type of genomic content that we have in microbes are plasmids. So we talked about this a little bit, but they are generally smaller than chromosomes. You can find them in basically all of the different microbes. They're usually circular, although if you remember that table from before, that there are cases where there are linear plasmids. So these need host proteins to replicate them. So you kind of think of them like viruses. They don't have their own polymerases or anything around, so they need to actually use the host's machinery to replicate. And they're not actually, the replication of these is not actually tied to the replication of the genome. So just because your genome is starting to replicate, that does not mean that the plasmid is going to replicate at the same time. So the plasmids have their own origin sequences. So they have their own ORI sequences, and that kind of controls when the replication of the plasmids happen. So they can contribute to the physiology of the cell. The most common example is they carry antibiotic resistance mechanisms. And again, they can be transmitted between cells, most commonly through conjugation. So plasmids can actually replicate through two different methods. The bidirectional method is essentially the same as you would think of for the genome replication. So, you know, it has an origin site and it'll go in both directions, or terminator sites and all that. The other way that plasmids can replicate is through this rolling circle amplification. So this is a unidirectional process. So this rep-A is the replication initiator for this process. It binds to the origin and nicks only one strand. So you only get one strand of the plasmid. So that's now coming apart, and so you can start replicating this one strand here. So rep-A will hold on to the 5' phosphate of the nick strand, while the 3' hydroxyl serves as the primer for the host DNA polymerase. So basically what that means is, remember the polymerase needed a primer, right? It needed someplace to start. And so if you only take off one strand, now you have that spot where there is actually that 3' hydroxyl that's already sitting there. So you don't need to add like another RNA primer in this case, because it's only created that one stranded nick, so that other hydroxyl already exists. So that already exists, and so now it can keep going. So because this doesn't have all the same machinery as the polymerase and the replosome, the rep-A also recruits a helicase to start unwinding, and then also the proteins to protect the single-stranded DNA also get involved. So it'll recruit the helicase so that this thing can keep unwinding bit by bit as it goes around. And so as you can imagine, based on what the name is, it's rolling circle amplification. So it's going to go and it's going to keep going in a circle all the way around until it makes the one circle. And once that happens, it's going to get rejoined by rep-A. So rep-A has that ability to rejoin that end. And now this single-stranded copy, so you'll have this one circular copy, right? It'll get protected by the SSBs until the complementary strand can be made. All right, so now remember we talked about how the replication of the plasmids aren't happening necessarily at the same time as the genome. And if you can imagine, because the genome is very, that whole process is synced with septation, and all the division and all of that happening is timed for the cell. But now if you can imagine, you have these extra circular DNA pieces just kind of floating around in the cell. How do you make sure that a plasmid makes it into the daughter cell? Because you can imagine that maybe the plasmid's like way off on this side. If there was only one plasmid in the cell, and it was way off on this side, and it was being replicated over here, but then the cell starts dividing, it's missed its chance to make it into the other cell, right? So in some cases, that's just, the plasmids just go by chance, and that's what happens. And if they make it, they make it. If they don't, they don't. Other plasmids, in that case, they go by probability. So they have, within that ORI sequence, it tells the cell how many copies of the plasmid to keep around in the cell. And so you can have high copy plasmids, so 50 to 700 copies per cell. And so just by chance, when the cell divides, it's a high probability that it's going to make it into both cells. In the case of low copy plasmids, so they have some other tricks that they can play. So this example your book gives from Salmonella for this plasmid R1. So the plasmid encodes for these separate genes. Essentially what happens is the par R and the par C, they form a complex, and it forms a complex with the plasmid. So then this par M, which is an actin-like filament, so it attaches to one of those complexes, and it essentially starts pushing the plasmids to the other end. So it grows and it pushes the plasmids apart so that you'll have them on the opposite ends of the cell, and so that ensures that each daughter cell gets one copy of the plasmid. So other things that it can do is the plasmids can actually carry genes that are beneficial to the host, so in a specific environment, so the host will want to try to keep it, so that ensures that they will make it on into when the host is doubling.
Can the plasmid integrate in the chromosome?
So every once in a while you can get some genes that will do that, but generally it's pretty easy for them to just hop along and go along in other cells. All right, so now it's we switch topics a little bit and kind of talk about a few different technologies that have come out of the different enzymes and processes that we have talked about so far. So the first one comes in with restriction enzymes and cloning. So we talked about one specific restriction endonuclease earlier when it comes to with the nuclease that will look for when it's coming in, it's methylating the different sequences. It'll only methylate that one sequence, that GATC sequence, and so when you start having these endonucleases come in, it'll recognize that methylation, and we talked about this a while back with the viruses, right? If it doesn't recognize that doesn't have that methylation, it'll look for those sequences and it'll just kind of trap them up. So it turns out that there are a ton of these restriction enzymes that come from, whether they come from different species or from the same species. So here are just a few examples. So what they do is they recognize sequences that are 4 to 6 bases in length, and there's always palindromic sequences, so they read the same forward and backwards. So if you read this one, so when you're reading the 5' to 3' side, so this one's AGCT, right? And then from this side again, it's AGCT. And so they always read the same forward and backwards. So they will recognize whether it's a 4-base or a 6-base sequence, and then they will cut that sequence. And they can cut it in 2 different ways. So it can cut straight down the end, like in the middle somewhere. So this one cuts in the middle, but there could be one that cuts here. So the ones that cut in the middle, it creates what we call a blunt end, because it's essentially just creates 2 blunt-ended DNA pieces. But there are other options where you have these that will create what we call sticky ends, so these guys here. So it's going to recognize this 6-base sequence here, but it's actually going to come through and it's going to cut this following this line here. And so what you end up with is kind of, let's see if I can draw this. So you end up with a DNA piece. So this part is double-stranded, and this part is single-stranded, and then this part here will be double-stranded. And so you'll have these 2 pieces that are separated now. So these ones can still bind to another sequence, so that's why we call them sticky ends. So what these restriction enzymes ended up being super useful for is what I'm sure many of you have heard of now, is this molecular cloning. But so the first people to kind of think about this was Stanley Cohen and Herb Boyer. And so this is like the first time that the words cloning was used in the terminology of cloning. You may hear of it as cut and paste cloning or restriction cloning, depending on what lab you're in or who you're talking to. But the idea behind this is, and why I like to sometimes think of this as cut and paste cloning, is because so you take the restriction enzymes that you're using and you create these different sticky ends. And you can actually create that on 2 different pieces of DNA. So in this case, so when Cohen and Boyer were doing this, what they had thought that they wanted to do was they knew about plasmids and they knew about how to cleave all these things. They thought, well, can we put a different DNA sequence into this plasmid? And so what they did was essentially use these restriction enzymes to cut both the plasmid and this foreign DNA. And so when you create the ends here, and if you cut them with the same restriction enzymes, you can create compatible ends. And so this end here from the plasmid is actually sticking to this end here from the foreign DNA. And so now you stick these 2 together here, and then you throw in a DNA ligase. So just like the one that closes up the end of replication, you throw that in there and it ligates it, and now you have a new plasmid with a new sequence in it. And so this method is very commonly used, and it's been used for many years now for cloning. And it's extremely useful. So most of your like molecular biology labs will probably clone lots and lots of different things. All right, so your book talks about PCR, but I'm gonna skip PCR. I'm sure you've all heard how PCR works and how important PCR is. I did want to talk a little bit about DNA sequencing technologies. And so we mentioned this back in the historical context about Sanger, who was the first guy that developed these sequencing methods. So there's this, a lot of these images come from this E-Appendix 3. So again, I will double check and make sure that that's up there if after class. So essentially what this does is uses DNA synthesis. So uses the same kind of mechanisms that we're using for replication to be able to tell us and actually figure out what the sequence of an unknown, so plasmid, or if you put in, you can even do linear DNA into these reactions. So the trick though is that instead of just doing, so you're going to have your let's say we're trying to sequence a plasmid. You put your plasmid into the reaction, and instead of just kind of copying the exact plasmid sequence itself, it's going to throw in these special nucleotides. So it's going to put in these dideoxynucleotides. And so what happens there is essentially it stops the replication. So it stops the elongation of new DNA at that point. So you can't add any new bases on there. And so what essentially you will have is, and they have the trick is that they put the dideoxys in at a very specific ratio to the regular nucleotides. And so you'll end up with a distribution of different size fragments that have stopped at essentially each space, each nucleotide on the thing that you're sequencing, just by chance. And so it was a lot of work for them to figure out the specific ratio of the dideoxys to the regular nucleotides. The other trick is that each dideoxy is tagged with a different fluorescent dye. So the A's, the T's, and the C's and the G's all come out with a different color. So that's why these here is what are called chromatograms. So they all have different colors. So then what you do is you collect all the different fragments that you have, and you essentially are running them on this huge gel. And so the gel separates the bands by size, right? And so every time one of the bands will stop moving, and then you'll have this laser and it'll see what color that was. And so you know exactly what base that was. And so you create essentially these chromatograms that have told you that at this position, all the fragments that I got stopped were this yellow, which is T. And so you do a lot of these and then essentially use a program to sort out and tell you what exactly which each one of these nucleotides are. So the limit to the Sanger sequencing was that you could only sequence about 1,000 base pairs at a time, and even that reaction itself would take a few hours. So the next big thing that by now it's been several years that people have been working on this, but it's what's called next generation sequencing. And the biggest, most well-known company is Illumina, and they do what is called sequencing by synthesis. I put the link for their, the Illumina video on there. It is kind of a very complicated process. I do not expect you to understand it. But it is kind of interesting to think about it, because there is a lot of physics and a lot of material science and engineering and biology that had to go in to making this system work. So it's just a good video just to watch to appreciate all the different technologies that has to come together for this to work. But basically, the end result is that this method can allow you to do about 600 gigabases per run. So, going from 1,000 to 600, granted, though, depending on which machine you're using and how much resolution you want on your sequencing, it can take a week for you to run that machine. But essentially, you can start, sequencing entire genomes, and the machine will just sit there and it'll do its thing, and then you have to pass it through some bioinformatic pipeline to actually get out what you need. But now the throughput for sequencing is way, 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 way bigger, essentially, and way faster. So how does this relate to microbiology, and why should we care, right? So this now enables new types of studies, which we call metagenomics, and so it allows us to essentially sequence microbial communities and understand what other microbes are out there. And so, all of those unculturable microbes that we can't grow in the lab, we know so much about them because of sequencing technologies, and we know that they exist and we know that they're there. And so beyond just sequencing genomes, there are new technologies out there now that's based on these next generation sequencing platforms that allow you to sequence all the RNA in a sample. It'll tell you things about protein interactions. There's just a lot of things that you can do with these different methods. All right, so that was the end of chapter 7. So just as a recap, so chapter 7, we talked about DNA, and remember about how the compacting the chromosomes and all that stuff was in chapter 7, and the replication and plasmids, and then finally some of the technologies. Okay, so with the last bit of time, I had several people email me with some topics that they wanted me to review, so I'm going to go over as many of those as I can right now. I encourage you, I still have office hours today. If you have more questions, or you can make an appointment with me for tomorrow. I have time tomorrow and Friday if you want to schedule an appointment with me. Okay, so the first question was help in understanding how to classify heterotrophs versus autotrophs. So if we remember about the different ways, the namings and the different things that we were going to classify microbes from, the idea was this for the heterotrophs versus the autotrophs is where do the cells get their carbon for building biomass. So this figure from your book, I thought maybe helped put it into a bigger context. So this is just like a simple version of a carbon cycle. And so essentially for the autotrophs, what you want to remember is that they use carbon from CO2 for fixation. So this is where they're getting their carbon, is from CO2. The heterotrophs are using the polysaccharides, so they can get them from sugars and whatnot. And that's so they're generally organic compounds, and so that's where they're getting their carbon. So they'll take these molecules, and they'll break them down, right, through these. So hopefully some of these processes look familiar, but we will talk about them later. But they will break down these molecules and use that as their carbon source. So if you remember, there are two other ways that we classify bugs, right? So based on their energy source. So what's also on here is so you can do that by light, right? Or by chemicals, which is not actually on here. But so maybe I'll write it down here. So you can get their energy from light or from chemicals. And then the final one was electrons, right? So you can get electrons from either from so inorganic molecules, which is the litho prefix, and then you can get them from the organic molecules, which is the organo prefix. So inorganic molecules, you know, they're things like hydrogen sulfide, ammonia, nitrate, things like that. And then your organic molecules, again, are your sugars. Okay, next topic, requested topic was the phosphotransferase system. So the phosphotransferase system is an example of group translocation. So I'll write that in here. So this was a method for passing sugars, getting sugars to get past the membrane, right, to get that into the cell. So the goal for this is that you want to increase the concentration of the sugar in the cell, because like we just showed, right, the cell's going to use that and break that down for energy and biomass and all this stuff. And so let me see. So if I draw this here, so we're going to do glucose. So let's see, G stands for glucose. And so the goal, so what we want, so this is the goal, is to get as much glucose as we can in the cell, right? Because the cell needs lots of glucose, right? So we want to get this into here. So now if you kind of think about passive versus active transport. So if we go from here, eventually we want to build up this store of glucose in here, and so eventually it's going to be that there's going to be less glucose out here than there is in here, and so this is going against its concentration gradient. Right? Essentially, anytime you want to move something against the gradient, that requires energy, right? It's harder for that to happen. And so what this phosphotransferase system does is it essentially does this trick where it modifies it. So what it actually does is, so if it's glucose out here, when the glucose goes in, as it's going in, it actually modifies this to glucose 6-phosphate. And so now what we have in here, is a bunch of G6P. And so now this kind of modification trick, so now there's actually not glucose in here, the glucose is out here, so now it's actually going down gradient. All right, so how does this actually do this? So what it's going to do is it's going to get this phosphate from PEP, so from phosphoenolpyruvate, and it's so it's a series of different protein complexes, and so we have. I should have drawn this one bigger. Let's see. So we'll get. What does what stand for? Phosphoenolpyruvate. So let's imagine that we get this. This is the phosphate bond that's moving off of from PEP. So there are 3 different enzymes in this system. Or proteins in this system. So it's gonna pass. PEP is gonna get. It's gonna pass this phosphate to enzyme 1, and so it's gonna. This one is gonna now have the phosphate, and that one is gonna pass this phosphate down here. So this is enzyme 2. So now this will have the phosphate. So it's this whole process. And then as that, now this phosphate here, so as the glucose comes in through here, this actually passes the phosphate to the glucose. And so this ends up as 26P on the inside. And the specialized thing about this is that, so you can have a single membrane that will have several of these things, but this one will only do glucose. So it'll have separate enzyme 3s. So this one will only do glucose, and then this one will do something like mannitol or something. So there'll be specificity for the protein in the membrane for the sugars.
Oh, it's a protein? I was going to say, what's the circle?
Yeah, so these are membrane proteins, sorry. Okay. All of these circles are proteins. Yeah.
So the membrane protein will add the phosphate.
So the one in the membrane is where the sugar's coming in on. So I'll just draw it again over here. So this is one protein here. There's going to be another protein here that eventually gets that phosphate. So the phosphate's going to come attached to here. It gets passed along a couple of times. So this one is sitting attached near the one that's in the membrane, and then as the glucose comes in, it passes by this, and the phosphate comes and attaches to the glucose. Okay. Any other questions about this?
So would that be like an exchange of energy then?
Yeah, so this phosphate bond is considered a high energy phosphate bond.
So then what's like the benefit of that rather than doing like active transport?
I don't know. So, in the end, it probably has something to do with the balance of how much PEP versus ATP. ATP is probably more valuable, more expensive.
So just to verify, enzyme 1 grabs the phosphate, and then as the glucose is coming in, that's when the glucose gets the phosphate, right?
So enzyme 1 gets the phosphate from PEP. It actually passes it to enzyme 2, and enzyme 2 is the one that as glucose is coming in, it'll pass that one to that. Go ahead.
Does PEP just flow around the intracellular matrix? Is it just floating around, or does it get transported out?
I'm not sure if it would get transported out. It's part of, like glycolysis and TCA and all that stuff. So the cell balances its concentration pretty tightly. So it's usually around. All right. So the next topic we'll review is the lytic versus the lysogenic cycles. So again, so these refer to bacteriophage, right? So this does not count for human viruses. This is specific to bacteriophage. And so you can have phages that are only lytic, and you can have phages that can do both cycles, the lytic and the lysogenic. So I'm just going to put this in the context of the kind of all those steps that we went through the the phage life cycle. And so the first step, right, is host recognition and attachment. And this needs to happen regardless of if it's lytic or lysogenic. This is the same thing, no matter what. The next thing is genome entry. Again, same. Lytic, lysogenic, doesn't matter. So now we'll kind of split this, and we'll just talk about the lytic pathway. And so this is kind of what you would think of as more as your typical pathway of what would happen immediately. And so the third step, right, is if remembering the overall life cycle process, is to replicate the genome. So now if we kind of remember, when we talked about the lytic cycle, we had what was called the early and late stage genes. The early genes were the ones to make the capsids and make all the proteins that the different virus particles needed. So that's part of step 4 in the cycle, is to make viral proteins. And then step 5 is assembly. Right, and step 6 is exit and transmission. Oops. So for exit, the late genes is a lysis enzyme, right? So this is your typical kind of virus life cycle, right? So all the different steps that like essentially every virus needs to go through. So, and this is what it would do for the lytic cycle. So now if we think about the lysogenic cycle, we can just, it's a nice way to think about this is an alternative pathway and extra steps. So for bacteriophages that are temperate, that can go through this cycle, it's essentially, they can pick which way they want to go. And so instead of just having it go straight from here to here, we'll call this 2A. And so it's going to integrate into the host genome. So this is where now it's called the prophage, right? And so things that happen here while it's integrated into the host genome, that actually prevents further infection by other virions. So once it stakes its claim, it's like, you can't take over my cell, this is mine. And then this is essentially a hiding spot for the bacteriophage in the bacteria genome. So it just replicates along with the host. And so then the next step would be, so this one is now, it wants to get out of the host genome, right? So it's excision from host genome. Okay, so this can either happen randomly, or in most cases it's going to be triggered by some kind of environmental stress. So essentially, if it senses that the bacteria is in trouble and the bacteria is going to die, that's no good for the phage, right? So it's going to want to get out of there and hopefully find a better host. And so it's going to excise itself from the genome, and one of the side processes that can happen, so it can pick up host genes at the same time. So that cutting process is not perfect, and so sometimes it'll pick up extra genes, which will then get thrown into the next cell that it goes into. So after it does that, it essentially slots itself back into number 3 here. So instead of going straight from 1, 2 to 3, we have it go from 1, 2 to 2A to 2B, and then at the right time it'll shoot back into 3, and then just do this whole thing.
when they pick up their host genes, isn't that how they actually like evolve to target specific cell types when they take those genes?
I'm not sure if that's how that happened.
I remember reading something about how like viruses evolve by taking.
That could be. I mean, that makes sense. It'll, because when it goes in, it's gonna replicate those other, and if it takes like a whole protein, it might actually make some of those proteins too. So, could be. I'm not sure exactly if that's the case. Any other questions about this? Yeah.
In the minute cycle, under early and late, are they both different process?
No, so it's expressing, so it's doing, transcription, translation of the genes. It just does it at a specific time. So it'll do the early genes is going to do the ones to make the viral proteins, because it wants to make as many copies of itself and get ready before it makes the late genes, which is the lysis gene. So it kind of just takes its time and it waits to transcribe the lysis genes.
Okay, so 4 is early and then 6 is like the late gene.
Okay, so we probably have time to do one more. So I'll go through membrane fluidity. So the question was essentially, when are the differences in membrane fluidity most important? And so when is it important? So the most important case is in the case of temperature, right? So you want to do so we will think of these things as in two different types of environment. We have your hot environment and your cold environment. So what happens in the hot? Just in general, what happens to molecules and things in the hot environment? So molecules are moving faster. And bonds start melting, right? And then the opposite side, in the cold environment, things are moving a lot slower. And you can get freezing of the different molecules. So in these two different environments, your average membrane what would happen to these? So your average membrane in a hot environment would become too flexible. And essentially, it'll fail to maintain the cell shape. So it'll fail to maintain the integrity of the cell. In the cold environment, so it'll become too rigid, right? You can start getting kind of freezing. And if you remember now, if you kind of go back and you remember the fluid mosaic model and how important it is for proteins to be able to move around in the membrane, that essentially won't be able to happen, right? So so no fluid mosaic model. All right, so what then do the cells need to survive in these temperatures? So if this is becoming too flexible, normally it would need to survive better, it would need a membrane that's more rigid. And in the opposite case, if it's trying to fight against rigidity, it would need a membrane that's more fluid. So how do we achieve these things? So things that will give us more rigid membranes, right? So we have the longer lipid chains. We have fewer double bonds. And we can add molecules that will kind of prevent and make things more stiff. So things like the hoponoids. And like you said, the planar, the ring structures. Okay, on the other side, what we want then essentially are the opposite of these things, right? So we want shorter chains. And then more double bonds, which is the same thing as unsaturation. And so essentially things that have kinks, right? That create kinks, so that creates space in the membranes, and so you get more fluidity. Okay, sorry I didn't get to some of the other requests, but again, come and talk to me today or tomorrow or Friday.